Welcome, everyone, to this afternoon's master lecture. It is my privilege to introduce Dr. John Cacioppo, Dr. Stephanie Cacioppo. John Cacioppo representing this afternoon's master lecture, which is co-authored by both. As many of you know, Dr. Cacioppo's research focuses on social neuroscience. In fact, quite literally invented the field, as well as on the study of social isolation, social resilience, and evaluative processes in emotions and attitudes. Dr. Cacioppo is the Tiffany and Margaret Blake Distinguished Service Professor at the University of Chicago and Director of the university's Center for Cognitive and Social Neuroscience. He's also a fellow of numerous professional organizations, too numerous to list here. He served as president of the Association for Psychological Science, the Society for Personality and Social Psychology, Society for Psychophysiological Research, and the Society for Social Neuroscience. Dr. Cacioppo's distinguished career has been marked by an impressive number of research awards. This is a tiny subset, including the Scientific Impact Award from the Society for Experimental Social Psychology, a Merit Award from the National Institutes of Health, the Theoretical Innovation Prize from the Society for Personality and Social Psychology, and the Distinguished Member Award from PsyChi. That one I really like. Dr. Cacioppo received the Patricia R. Barkas Award from the American Psychosomatic Society, Distinguished Scientific Contribution Award from the American Psychological Association, Campbell Award for Distinguished Scientific Contributions from the Society for Personality and Social Psychology, the Award for Distinguished Scientific Contributions from the Society for Psychophysiological Research, and the Troland Award from the National Academy of Sciences. That's not all of them. I've just run out of time. The title of today's talk is New Developments in Electrical Neuroimaging with an Emphasis on Individual Differences. Please join me in welcoming this afternoon's master lecturer, Dr. John Cacioppo.
Thank you. It's a real pleasure and honor to be with you this afternoon. I want to thank Dr. Mortensen for that very kind introduction. You know, he's an extraordinarily capable man, but I want to give him special thanks for the wonderful late winter weather that we're having in Chicago. I hope you're enjoying it. It's not typical. So thank you very much for the wonderful weather. We're going to be speaking today about electrical neuroimaging. Now, electrical neuroimaging is something that goes by the name of EEG. Electroencephalography, but you'll see why it's taken on this slightly different term now. And I hope to, in the course of the talk, convince you that over the last several decades, we have learned how genetics can contribute to our understanding of individual differences in personality, but that also the study of the brain, and in fact, not just functional MRI, but electrical imaging can also tell us important information and allow us to test hypotheses about individual differences and personality processes. But to do that, I want to put it in a broader context. So as you probably know, electroencephalography was developed, introduced in humans by Hans Berger, a psychiatrist in Germany in 1926. It's a very simple, crude recording. It was a single electrode at the vertex of the scalp. And as you know, 1926 was a very long time ago. Scopes was found guilty of teaching evolution in the United States. William Jennings Bryant was on the prosecution, and Darrow was his defense attorney. It was one of the few cases when he actually lost, and this trial was famous not only because of the topic, but because of the two lawyers who were hearing this case. So that's a long time ago. If you move forward to the 1970s, we were doing EEG from multiple recording sites. But our computer technology, of course, was very primitive. In fact, I had a lab, I was doing EEG in the early 70s, and it didn't look too unlike what you see here. All the scoring was by hand. You looked for waveforms. When you did have something relatively sophisticated, we came up with some early computers that we used, with paper punch tape, no less. We used it to study affective processes and individual differences, but those measures were extraordinarily crude. It was basically the alpha power that you saw, how much alpha activity, which was taken as the inverse of how much activity the mind was actually engaged in during that period. And we were fairly sophisticated, so we talked about how much activity was going in one hemisphere versus the other hemisphere. By today's standards, extraordinarily crude. But it is somewhat noteworthy that when you look back those 30 and 40 years, we were able to cull some interesting information about individual differences from those measures. Now again, that too was a long time ago. That was the period of the Falkland Wars, where Britain and Argentina had clashed. And so we can move forward a couple decades. And during this period, event-related brain potentials had developed. The reason event-related brain potentials, for those who aren't familiar with this, was interesting is normally EEG is plus and minus about 50 microvolts. That sounds like a very small number, but when you magnify it, it turns out to be quite a wide range. And eye blink, by the way, is about 250 microvolts, so you can see how these signals are still relatively small. But if you time lock your EEG to some stimulus or event, and you average many, many such tracings. Let's say I'm just looking at a sound, or I just am presenting a sound. An auditory evoked potential is what that would be. You don't know exactly when that sound is going to occur. The way in which we record EEG, it's distributed around 0. So any activity that's not processing that stimulus, it's not time locked to that stimulus, sums to 0 or close to 0 plus noise over the course of many trials. And what this slide is showing you, the more trials, the better you can see the components that are linked to the processing of that particular stimulus. Well, that can be interesting in terms of individual differences, because we often think different individuals aren't thinking identically. That's what makes the individual differences interesting. And so you can test specific hypotheses about how they're thinking differently using such technology. You just need the right task. Well, in early research, even when we had a large number of recordings, people picked their favorite site and they picked their favorite peak or trough. So it was still relatively crude. Notice you use computers for recording and for averaging, but for analysis, you can do this by hand. You're just measuring the peaks and troughs, the amplitude. Sometimes you do the area instead of just the amplitude of the peak or trough. Again, it proved important and informative because it did allow you, with appropriate tasks, to test hypotheses about specific processes. Like a functional neuroimaging, it's important that you have the behavior down right. You know what's going on behaviorally before you port them into any kind of neuroimaging lab, and that was clearly the case here as well. Well, the 90s were a long time ago as well. If you remember, this is the time of Janet Reno and the Waco tragedy. Well, let's move forward at least to the new millennium. We have functional neuroimaging that's made an important presentation of itself onto the field of behavioral sciences. And we started to look, people generally started to look at all kinds of differential activation patterns in the brain as a function of tasks or individual differences. It turns out 2000 was a surprisingly long time ago. If you look at these pictures, both George Bush and Al Gore look relatively young. And you may recall the Florida debacle that turned out to lead to George Bush being elected president and all that followed. Well, there were problems with that election that we could complain about, and there were problems with fMRI that we've come to recognize. And those are that the temporal resolution is poor. A lot of times, one of the things Donders gave to psychological science is timing matters. What goes on and how long it takes matters. And we lose a lot of that information with fMRI. It's a little bit like what you get out of your iPhoto program. It shows you all the photos that you took during the trip, but it doesn't really give you an idea of where you were and when you were there. So it's ordered in time, but you don't have much temporal information. It's only ordinal information at best, and that's better than you get out of the fMRI. The cost of the infrastructure was very high. It's almost impossible, as we've all come to learn, to support a magnet and all of the physics and equipment that goes with it with research users only. It takes clinical users to actually pay for the device. The cost of scans are high, so it makes way for underpowered studies. And it turns out underpowered studies leads to both type 1 and type 2 errors, so much so that the likelihood of having an observation be accurate is about equal to the likelihood of having it be inaccurate, which has created a lot of chaos in terms of trying to make sense of the data now. I mentioned this timing. Well, if we wanted to recapture the millisecond precision that we have in reaction time, then you're forced back into looking at EEG or MEG. And so Stephanie came from medical school in Switzerland, University of Geneva Medical School, and she was doing high-density EEG, and she was looking at EEG in a completely different way. What she did basically was to look at, say, 128 channels, and she looked at them at the same time. Now, when you do a study of this size, let's say 128 channels, we call them vectors, you'll understand why in a moment, and a 500 hertz sampling rate, we actually use 1,000 hertz, let's say 500 trials, a 500 millisecond baseline, 1,000 millisecond task window, and you run 50 subjects because we don't want underpowered studies, you get 2,400,000,000 data points. This is not something you're going to do by hand. You're not even going to ask your graduate students to do this by hand unless you have many generations of graduate students to work on this. So here's where EEG analyses and where electrical neuroimaging starts to emerge. By the use of computers, and we're working with the supercomputer group at the University of Chicago on some of this, we're able to find information that we've just not been able to deal with previously. There is order in the data, but there's a lot of data to deal with. So the idea here is to look at all 128 sites simultaneously, not to pick your favorite site, to map from that these scalp distributions. So now those 128 vectors or channels represent a high-density sensor space. That sensor space represents the electrical signals that are emanating from one or more nuclei in the brain that are active. It's called the forward problem. If we know what those areas are, and we know the conductances, that's where the ventricles are, where the white and gray matter are, and we have good hints about that, and we can always do MRI and determine that, then we know we can predict accurately what the scalp distribution will look like. All right? That's not the hard problem. The hard problem is the inverse problem when you try to go the other way. All right? So that's basically what you're doing, is you're using this high-density sensor space millisecond by millisecond to look for when is the brain stable, when are there certain neural loci that are operating, when does one or more of these loci change, because if just one of a set, it's like chemistry, right? If one of them changes, you have a new substance, and you'll have a new configuration. So you're looking for when is it stable, that's called a microstate, and when is it not stable, when is one or more moving, and then you reach some new stability, which would represent possibly a new set of neural loci, and I'll come back to that particular point. So you have, basically, to summarize, you have a subject, you're going to do 128 channels. That's what it looks like if you take all 128 and you map it across a single trial. You get this mapping of electrical distributions across the scalp. Now, I said you get that for every millisecond, so this is what the data actually look like. If you're looking at it in this sensor space, and what Steph and others had been doing at that point in time was doing cluster analyses or K-cluster analyses or hierarchical cluster analyses. Basically, they're looking for when are these maps correlated with each other? Where are there periods in which they're similar, okay? And you can see, you can reduce this large set of data to a much smaller set of stable, or at least similar states. And these are called microstates. And this analysis was really begun by Lerman and colleagues in the 70s and 80s, if you can imagine. Now with higher density space and better computational powers, we can get a lot of information from this particular approach. There were some issues with this mathematical approach, though, that became evident over the last several decades and bothered Steph and myself in particular. They include that these clusters that get identified, you often see the same clusters at different points in time. So if I go back to this slide, you might, it isn't this case, but you might see this map and this map be very similar. But they treat them, and mathematically they are treated as similar, right? But that's not how they're treated by the investigator. They're treated as if they're discrete different states. And the reason is, when we're looking at event-related potential, something that occurs at 100 milliseconds and something that occurs at 500 milliseconds, we think are qualitatively different. And so despite the mathematics, they weren't treated as if they were the same process. Well, I'm a math geek. That bothered me. If they're mathematically equivalent, that tells me the process is equivalent, the underlying neural source is equivalent. And so that was a potential problem. We wanted to kind of look at an alternative mathematics to see if that were an artifact of the quantification or it were actually something more reasonable. There were no transitions. You go from one microstate to another microstate to another microstate. Well, I don't know about you, but I don't pop from one location to another location. It takes me time, especially in Chicago, to get from one place to the other. And what we know about brain activity from single cell recording is that it takes time if you're moving forward. It's not unusual to see activity, say, from a visual stimulus, hit the visual cortex, move forward, and the activity in the forebrain or in middle regions feed back to the visual cortex and tune how much you're looking at that stimulus. We've seen that in our own fMRI research. All right, and in single cell with animals, that's been documented, that you get this recursive neural processes going on. But it doesn't pop immediately. There's a transition period. It can be 5, 10 milliseconds, but there's some period of transition. And homogeneity, as in all of ERP research, has been assumed. Now, that's a real concern for people with your interest and my interest, because basically it's saying everybody's head is the same. So we can use the average, and now tell us what everybody's thinking. Well, I'm a big believer in individual differences and using that individual variation as theoretical information, not error variance. And this is treating it as error variance, all right? And so we wanted a technology that would allow us to look at that, quote, error variance and test whether it really was error or whether, in fact, there was signal there. And if so, to make suggestions, so inductively, we have information that might allow us to see individual variation that we hadn't saw previously. Why? Because the brain's doing something different. Maybe the phenotype, the psychological processes, and the individual differences are actually going to be informed by that variation. So what we developed was a set of mathematical operations, not a single one like K-cluster, but a series of mathematical operations that we now apply. And you can see some of the steps, but let me just give you a quick tour of the mathematics. This is just pictorial. I'm not going to bore you too much, and then I'll get to how it might actually be useful in telling us what the mind is doing. But what we used was based on video compression algorithms. In video compression, with high-density TV, they're not sending a complete high-density array of information to your receiver, TV receiver, every 17 milliseconds. What they're doing is they send a set, and then any pixel that changes, they refresh that one. So that's all you get. There's a lot less information that you have to send when you do that, okay? And so that's a video compression algorithm. So what we did was to use the mathematics of video compression, and we used the baseline, and there's a special way to do baselines, so we developed a model of noise in the brain. Now, I say noise in the brain, it's actually noise in our recording system. What's the baseline variation that we're seeing when there isn't any time-locked psychological process going on? So what that means is we're using the baseline and we have jittered the baseline. So they don't know when the stimulus is going to appear. It's not that they're not doing anything. It's that nothing they're doing is time-locked to that stimulus because they don't know when it's going to occur. So that means over many, many trials, it should move towards zero. What's not at zero is our model of noise, all right? So if you will, it's our model of the measurement of brain activity that represents simple noise. We use that, we create confidence intervals around that, and then we predict forward. If it's a stable state, what I see now should be present 10 milliseconds later. And so we do what's called a root mean square error analysis, which is what you see here. And when that error in prediction exceeds a certain threshold, then we have a new state, all right? It's stable, it's stable, it's stable. We go from A to B, and then when it starts to go down, we have now error again increasing, all right? And so you go up and down in this fashion. So A to B is a transition, B to C is a stable state. And if you look at the bottom line, what we've done here is we've got simulation. So I have a scalp distribution that's homogeneous, it's stable. I then take a simple linear transition from state 1 to state 2 as a stable state. And what you see is the mathematics of this showing how long that transition period is and how long the stable state is. Now, I can do transitions like this all day, and it'll sit and map up and down. So it's actually fairly simple in terms of the statistics. We simply establish, we usually use 99% confidence intervals, and when it goes above or down far enough and then up far enough, then we're entering a new transition state and stable states. Well, notice the mathematics all of a sudden introduced transition states. We can time how long it takes to get from one microstate to another microstate. Now, that gives us what we call putative stable microstates. These periods like this, like this and like this, all right? If I have a single neural locus in the brain and I increase the activity or I decrease the activity, that might happen because I'm an anxious person, I'm about to meet someone, and the closer that comes, the more my amygdala is getting activated. There's no real movement, let's say, hypothetically, it's just getting more active. That's gonna change the scalp configuration, all right? So how can I tell? That will look like a different microstate. It's not a different microstate, it's a change in how much activity was coming from the same neural locus, all right? So how can we tell that? RMSE is not going to tell us that. So what we do is we then go and we express it in a 128-dimensional space. Now, that's a challenging thing to kind of show you. So I'm going to show you in 2-dimensional space in a quarter of the domain, all right? So this is just one quadrant of 2-dimensional space. And these microstates, if you notice, are multiple periods long, all right? I illustrated it here with each microstate being 6 milliseconds or 6 recording bits long. Each one of those has a map. Those maps are not all identical, of course, because there's error. And so you have all the individual microstate measurements, and then you can calculate an average. We call that a template map, all right? It's our average. And then around that, we create what we call a confidence cone. It's a confidence interval in a 128-dimensional space, all right? If you know vector math, you know this is the case. If I have the same configuration in vector space, that is the same neural locus, but I change the intensity, that vector length increases or decreases. If I change the actual neural locus, the angle changes. And so what we do is we check every microstate that's identified by RMSE. The next one we say, does this template map fall within the confidence cone of the prior microstate? And we have seen it in our research both ways. Usually not, but sometimes it does. We have one checkerboard study where people were motivated. If they saw a 5 at a particular, in the fovea of that checkerboard image, they would get an extra $5. And what we would see is a lot of activity out of the visual cortex late. And then we saw in the first a change in state. In this, it wasn't a change in state, it was a reduction in activation. And what happened was they had looked, they didn't see the 5, and so they were still looking but not searching any longer. So you got less visual cortical activity. No change in configuration, neural locus was the same, it was simply less looking. All right? So that's the kind of distinction. That's why we actually go to this vector space. Okay, let me give you a simulation. We did lots of simulations. That's one of the nice things about supercomputers. You can run trillions, literally trillions of simulations. So we have here a homogeneous, calm state. We then change, this is a transition, change to a frontal negativity, all right? We change to occipital or posterior negativity in this scalp distribution. Very simple. Now what we're doing is we're adding a lot of noise. That's all this does. It's just random noise added to those channels, all right? So it looks basically the same, not just clean. And then we average. So we're running many trials. This is all simulated. We run many trials and we average. So here's your ERP. And you see the scalp distributions are cleaner. They look more like the original, but not quite as good. We run it through our pipeline, and a lot of this, what we've done is we have developed software and scripts now that do most of this for you. It even does the statistics and just gives you the output. And we did this because it's kind of complicated. We have a tutorial that's also been published on this, but it can be kind of complicated and it's easy to make errors. And so we just automated it. You don't have to do it in an automated fashion, but that's what this demonstrates. And what you get out is something that's pretty simple. This top set of terms here are the microstates. Remember, we went to an anterior negativity to a posterior negativity. Here you see two microstates. There's one here, ends here, one here, ends here. That's exactly what we simulated. That was a signal in the top channel. These are the microsegmentation. It gives you the stats on when it occurs, the transition periods, the duration of the microstates. But remember I said I don't like to assume all brains are the same any more than I like to assume all people are the same. So what we do is we do bootstrapping. We randomly select, let's say, 70% of our subjects. And we do this again. And we do it 1,000 to, in one case, we were looking at 43 trillion times. If I do all possible combinations of a large study, you literally end up with 43 trillion combinations. And you can only do that with supercomputers. So that's the kind of thing we've been playing with. This is 1,000 simulations of this, all right? And we calculate, here's what you see on these simulations, okay? These are bootstrap results. And what you see is a lot of homogeneity. Well, this is a simulation. This is only noise. There is no variance, all right? So you would expect this. And we've created this metric, plus and minus 10% of the time in which the aggregate, the grand mean microstates start and stop. And we do that because error, temporal error, only adds as you go forward. And so the later the potential, the more error you would expect. So we do 10% of the time. And this is the percent of our observations that fall within that plus and minus 10% of the time, okay? When it looks like this, and this is like 90 plus, it's a high 90% that's within that interval. When it looks like this, you don't worry about individual differences that you haven't quantified. Now, this may be one condition in an individual different study. But if there's additional variance that falls out, you have to worry about something isn't homogeneous as we thought it was. And indeed, our checkerboard study that I mentioned where that was motivated, we did that because we actually did the study, found variability. We have University of Chicago students. They're really geeky. And we realized, looking at the bootstrap results, there's some students who are sitting there looking at the checkerboard like everybody else does. We have some who are sitting and studying the checkerboards as they switch. And indeed, when we ran the motivated viewing condition, it looked a lot like the variability we'd seen. So that's the kind of value of these bootstrappings. You don't assume homogeneity. You actually can start to look and hypothesize what individual variation might be contributing to these signals. Well, let me show you a real quick demonstration study, and then we'll go to some studies of individual differences. I mentioned we did a checkerboard study. For those who've never seen this, it's a checkerboard. And every 500 milliseconds, the black squares turn white and the white turn black. So and we do this 252 times. It can be a fairly boring thing. There's no real cognitive requirements of it. And what you see here are very typical looking grand mean ERPs over O1 and O2 visual cortical regions. All right? This is the traditional way in which people have looked at this. And you see these bumps. These first three bumps and troughs are what's typically published in the literature. That's what people have focused on for a long time. So they may have 128 sensors. They're going to analyze 2. And they may have, 1,000 data points over the interval. They're going to look at 3 bumps or troughs. That's it. That's the standard method. So we did the same study, except we have used the approach that I just mentioned. And you see, we get, in fact, something that in the microstates that looks a lot like the 3 bumps that are typically looked at, but then we get 2 additional microstates. And indeed, this 4th microstate was not, there's no morphological peak or trough in the visual cortex. When we do, and I'll show you these results, when we do inverse analyses, it turns out to be a parahippocampal region. Now, that had never been observed in a human fMRI study. And it was short. It was only about 20 milliseconds long. It's one of these recursive things that occurs. Now, it could be that we're wrong, except Steph found a study in monkeys with high-strength fMRI that's in a visual checkerboard design that identified parahippocampal activity as one of the areas stimulated by the checkerboard. So that's the kind of thing that this technology provides. So let me just summarize real quickly. We have these mappings. We get high-density sensor space. Where we would like to go is to be able to make inferences that I just made about what is likely to be going on inside the brain. And I mentioned earlier, the forward problem is not a very difficult problem. All you have to do is have a good mathematical representation of conductance pathways to the sensor space and dipole orientation, and you can get, you know what the configuration is going to be. It's going from the configuration back that's bad. I actually was doing some of this in the 90s, but I never pursued it because you could put any dipole, any configuration, if you wanted to find a dipole, almost any reasonable dipole that is assumed source would fit the data. And that bothered me, the fact that I might put it where I theoretically thought it should be, and I'd get evidence to support it. But if I put it in an area that I didn't think it should be, that also supported the hypothesis. So to me, that was non-diagnostic, so we didn't pursue it. What Steph brought in was 4,000 dipoles in the gray matter of the brain. Okay, that's a pretty densely packed set of dipoles in the brain. And now it's not allowing you to pick and choose. It's going to test all 4,000 at once. That approach has mathematically things going for it. But there were some problems with how one identified the period. If you're just taking a peak or trough, you have one data point. And that's not a very reliable index, even if it's a peak or trough, right? Or you were using the microstate based on K-cluster, and notice they weren't identifying transition states. So remember that transition state that I showed you on the very simple first graph? Half of that transition state would have been seen as the first microstate, the other half would have fit into the second microstate. If I've got transition states in my microstates, my inverse solution is going to be poor, because I'm not only mapping where it was, I'm also mapping where it's going. And so they, in fact, in that field had already found using that whole period, the average didn't give them very good results, so they took the highest value. And again, anytime you have a single value, you're dealing with a questionably reliable measure, and that's going to hurt your inferences. So this is just a set of reasons why the microstate approach that we developed actually gives us better estimates of when that stability is. Therefore, you should get a better solution given the math is equally good. So this is just a handful. I've highlighted some of them, like it distinguishes between a change in the amplitude versus a change in the configuration, the locus of the neural source. When you do that, you get a map something along this. So these are the checkerboard results, all right? And not too surprising, these very early components are visual cortex, which has been observed not too. But then you get this movement back and forth. So it ends in the visual cortex, it started in the visual cortex, but there's a movement to the anterior regions before it snaps back to the posterior regions. Okay, let's go to individual differences. Something that Stephanie and I have been interested in for quite a while now is the individual difference of loneliness. The reason I started studying loneliness was because of the field of social neuroscience. We had conceptual reasons to look at it. There were arguments we had made. But I realized if we wanted to show that, I mean, I was in bio and social during this, right? And I realized, because my bio colleagues kept saying, you know, I'm uncomfortable when we go outside a cell membrane. Anything about outside the body can't be neuroscience. All right? You're going, why can't it? We're social species. We, by definition, create superorganal structures, and we've evolved superorganal processes. Those have genetic, cellular, hormonal, and neural mechanisms underlying them. They're not magic, right? So we can study those just like we study anything else. And so they would kind of get that point, and then they would forget. And literally, I'd have colleagues who say, 6 months ago, I remember you convincing me, but I'm not convinced. Remind me of why this is the case. So I realized that logic wasn't enough. And I thought, so let's look at something, that will, an empirical program of research that will show them how it's done. And because I wasn't studying loneliness before that point in time. And it wasn't that I sought out to study loneliness, I used an approach that's common in the neurosciences. If I want to study what a gene does, I create a set of mice, and I create clone mice who have everything except for one gene, that gene that I'm interested in. Now notice, I'm looking at this knockout set of mice, not because it doesn't have the gene, but I want to compare what it shows with the mice who do have the gene. So the focus is on the mice that are deficient, that gene. But you're doing it so that you can understand what that gene is likely to do. If you know the story of Phineas Gage, we learned a lot about the orbital frontal cortex because we knew his behavior before the tamping on obliterated his orbital frontal cortex and after. We're not actually interested in the whole. Whoa, sorry. I don't want to connect to box, so that should be okay. Okay, good. We're not interested in the whole. We're interested in the changes in behavior so we can learn something about the tissue that occupied that whole. So you know where I'm going. We studied social isolation, not because we were interested in isolation per se, but because we were interested in what the social brain was actually doing. Now, we looked at objective isolation, but we also, being psychologists, know that perceptions matter. And objective isolation and perceived isolation are not at all the same thing. In fact, they're very weakly correlated. And so we measured both, and the assumption was the brain is the organ for organizing our social world, our dispositions. And so if we looked at the brain's perceptions, that would be more valuable than just looking at the objective facts, and that's exactly what we found. And over the years, we developed this particular model. And I want to just point to one particular thing, and that is when you isolate an individual, they're explicit. Above, that is, in consciousness, they want to reconnect. What they're unaware of is that they engage in this hypervigilance, pre-attentive processing for social threats. They're doing it all the time, right? If I'm pre-attentively looking for social threats, I'm going to see them. I'm going to see them when they're not even there. It's just an ambiguous circumstance, all right? And so that was part of the model that helped organize a lot of data. We argued that the brain basically goes into self-preservation mode. We've done reviews of the animal literature as well, both the neurological and the hormonal, and we now have an animal model. We just published something on the genetics of this. And this hypervigilance is there throughout mammalian species. It's not unique to humans at all. I think the reason we're so unaware of it is because it's so phylogenetically deep. It evolved long before consciousness did. And so we wanted to test this notion that the brain is actually engaged in hypervigilance. We had behavior, but behavioral differences are hard to make a convincing argument, but it's pre-attentive. No one has any access to it. So we did a couple of electrical neuroimaging studies. I'm going to show you one. I'm going to show you one. It's which of it? The top one. It's using the Stroop task. And you know the Stroop task, right? Just name the color in which the letter string occurs. So if you will, please just try to do the next column. You know what this is going to do to you, right? It's much harder when they're out of color than when they're incongruent than congruent. And the interesting thing about this is you can't stop it. If you know this, you can't stop it. It's harder to name the colors, all right? And most people, you're obviously aware of it, but most people are completely clueless that their brain is doing this, right? So it's nice to illustrate the mind is doing things that consciousness not only is unaware of, but has no control over. Except we didn't do just color names. We used social and non-social words. And we didn't mess around with negative words. I mean, they were strong social and non-social words, all right? And we simply time-locked the presentation of these to our electrical neuroimaging and event-related paradigm, okay? Our non-lonely people showed a differentiation of the negative social and the negative non-social words after about 500 milliseconds, all right? The first 500 milliseconds, there's 5 common states. That meant that the non-lonely individuals, whether it was social or non-social, they responded to those negative words similarly. They were still processing those words. That's what those first 5 states are about. But they're not, the processing doesn't differ for the social and non-social negative words, okay? The lonely, in theory, should show that separation much earlier. And there are the data. Within 280 milliseconds, the brain has started to differentiate those words. Now, the other study that I'm not showing you is with pictures. That was even earlier because pictures were a little simpler for the people to process. So in both, we have this conceptual replication of lonely brains are distinguishing between social and non-social negative words very quickly, quicker than non-lonely. The advantage, of course, of the electrical neuroimaging is we can also do this inverse inference where we gain hypotheses about the neural origins. These are what you see from kind of the overall mapping, but if I just delve down into it and talk about what is actually different here, and I just did one microstate. You actually get long lists of these areas for each microstate. But if I just do one microstate, these are our high lonely subjects, these are our low lonely. What you see is in both high and low lonely subjects, social words, negative words, actually evoke, once they separate them, evoke more complicated sets of neural mechanisms than do the non-social. Social stimuli are complicated. They draw a lot of attention, and they require a lot of information processing. Even neutral expressions can mean a lot of different things. So people are kind of surprised by how much attention social stimuli, how much processing is actually going on. What's interesting to us was, of course, if you look in more detail, there are some common areas, as you would expect. Notice that lonely people are doing activating these common areas much earlier. But in addition, where it differs is there's more frontal regions and there's more somatosensory cortices activated in the lonely than the non-lonely. That is, there's a lot more, if you will, integration of what it is that they're seeing, and there's more emotional involvement. People aren't feeling that emotional involvement, but the somatosensory cortex is representing that kind of engagement of this organism by these negative social stimuli. These are stimuli that have real relevance to a lonely individual. The last study I'll show you is we asked one other question. And in the series of studies we found, among the differences we find between lonely and non-lonely is this hypervigilance for social threats. We see poor impulse control. In animals, it's called prepotent responding. All that means is animals are more likely, when you isolate them from a conspecific that they like, that they prefer, and you do that with partner preference tests, they show a pattern of activity that is characterized by responses higher in the response hierarchy. That's why it's increased prepotent behavior, all right? We call that increased impulsivity. It's the non-practice behavior. It's the things that we have to use self-discipline to achieve. Those get sacrificed in lonely individuals relative to non-lonely individuals. And what we did was we looked at we're back in the functional MRI setting. We looked at what do these lonely and non-lonely people show at baseline when they're just laying in the magnet, no task, no nothing. 55 subjects who vary naturally across the dimension of loneliness. And there's a series of calculations that are performed, but basically you end up with networks. And there are 2 networks depicted here. The one in red is the singular auricular network. It's been identified in cognitive neuroscience as the alertness network. That's the network that if I put you on high alert, or you're surveilling for something, you show increased activity in where those red rods are pointing, okay? And, this is Elliot Layton. He's the student who's been actually a super champ in working with us. These are not, the manuscript's going out after we review it for the last time. So these are very, very new data. But you see, that's this implicit hypervigilance. By the way, we find lonely people show less sleep salubrity. They show more micro-awakenings. In mice, they show less slow-wave sleep. So there's this process that we're not controlling that's activated, and you see it at baseline in lonely individuals. It's this increased alertness network, the singular auricular network, that's active, okay, and that's supporting all of those behavioral effects that we see. What you see in blue, so the red is positively correlated with loneliness. Lonely people show that more. What's in blue, they show less. And what you see is the supramarginal gyrus connected to the dorsolateral prefrontal. That's your self-regulatory or a self-control mechanism. And lonelier people, I mentioned, show poor impulse control. Animals show more prepotent responding, that is, poor impulse control, if we could talk about animals in that context. And you see this regulatory mechanism at baseline is degraded. It's not as active in the lonely individuals. So that's a kind of individual difference in mapping that you can get between these baseline assessments, connection assessments, and the EEG. So just to bring it around so that we can talk about this for a moment, the study of individual differences, the use of these, the brain and the genetic bases as signal rather than noise, allows us to see processes and test hypotheses in ways that are much more convincing, much more direct tests of a particular process than we could otherwise achieve in normal behavioral studies. So behavioral studies lead the way. They're the things that lead to the hypotheses, but the brain and the genetics can give us confirming evidence, and in the error in those, it can give us new hypotheses about what those additional processes might be. Thank you very much for your time. I'd be happy to answer questions. Yes.
Thank you for a very complex thing. I know I can't understand hardly any of it, but I'm doing the best I can, and thanks again for explaining. I have a question. The red network, are the lonely people who are hyper alert to loneliness?
No, they're hyper alert, period. So any social threat, they're going to be more reactive to. They'll pick it up faster.
Okay, are those connected to particular personality structures?
The personality characteristic is lonely. Loneliness is stable. It's about, over a 5-year period, it's about 0.7 temporal stability.
Okay, if loneliness is, if alertness is connected to more aggressive personality structures, that doesn't seem to fit into the idea of lonely because the progressives are lonely.
No, so one of the nice things about animal studies is you can randomly assign animals, so you're not studying animals who are lonely because they're aggressive, right? And what you see in animals as well as we see longitudinally in humans, so we have this 10-year longitudinal study that we've done, so we see the same thing in our humans. If you get lonely, you want to connect with others, but there's this implicit surveillance for threats. And so it doesn't take long when you find something that someone else does that you don't like. And so you actually turn out to be more irritable, a little more hostile. And animals, isolating animals is one way they actually institute study behavior that's aggressive in these animals. So the same randomization leads to the same phenotype.
So could it be said then that aggressive people are lonely?
Well, that's an inverse. And so A leads to B. B doesn't necessarily lead to A. Is Donald Trump lonely? He certainly seems aggressive to me.
He'll be alone.
He may well be, but as we know, being alone and feeling alone isn't the same thing. All right? So I think what's interesting is loneliness leads to aggressive behavior, but it's not the only cause of aggressive behavior. There are many other causes, which is why going backwards can be so ineffective. It can be caused by a lot of things besides the instrumentality, the emotions that are stirred can bring out aggressiveness in people above and beyond how lonely they are. Okay? But if I might point out, if I'm hypervigilant for social threats and I have poor impulse control, that's a toxic mix if I want to control my aggressive behaviors towards someone. So let me come right back to you, I promise.
Yes, Corey?
Yeah, this seems like a wonderful research. It seems to be that one could then make a design where you could do, for example, neural feedback with trying to train someone socially anxious and lonely more towards your template of someone who is not.
Yeah, no, it's an interesting idea to try to activate or see this system and try to then train them to lower that activation. That's never been tried. I mean, I think it's a really interesting idea. If one could do that, it would be fabulous because it's actually really tough to operate on implicit processes, right? It's not something they have control over. So I actually really like that idea. No one's ever done it. I've never even heard someone suggest it. Great idea. I promised to come back to you. I apologize. Go ahead.
Here we go. Could it be that the people who are hyper alert are lonely for some particular person? And is it that they're missing their mother?
Oh, look, you can be lonely because of a particular loss. Absolutely. The interesting thing is when you feel isolated from others around you, you become hypervigilant for all social threats, not just that. And there's an adaptive utility to it. It's self-preservation. You become, if you can't trust, if there's not mutual aid and protection, then your brain goes into a self-preservation mode. Remember, Caesar died at the hands of a putative friend. It has adaptive value. If you don't feel like you can trust others, maybe you can't. And making that error leads to a potentially fatal assault. So there is an adaptive utility to this implicit hypervigilance. We see the same thing in fish. We see the same thing in mice. We see the same thing in rhesus monkeys. It's actually stunning how similar some of these processes are. But I think it's because it has such an adaptive value. You and then you, okay?
So you mentioned the other species who show this same reaction. For them, you're looking at actual aloneness, I assume.
No, that's a great question. The animal literature, interestingly, they've studied isolation for decades, not because they were interested in loneliness or anything like that. They used it as a manipulation of environmental enrichedness. Sometimes environmental enrichment means they have toys, they have running wheels. Often, it means they have conspecifics present, okay? And if you look at that literature, it's kind of messy for two reasons. They're small sample sizes, remember what the problem of that is, and then they also don't distinguish. They do just separate. And Steph, actually, while we were doing the first review of the animal literature, realized that, wait a minute, sometimes they're separated from a preferred partner. Sometimes they're separated just from a strange conspecific. When you sort the data in that way, you see profoundly different results. And so it's actually important to know whether they're being separated from just another animal or are they being separated from a preferred partner. The latter give you very different results. It looks like our lonely subjects. From just other conspecifics, no, it looks like they're alone, basically. Okay, yes, please.
Is there a timing to these findings in terms of how long the animals have to be separated in order to identify?
Yeah, it depends on the animal. Usually it's about 2 weeks. So in the mice study, which is there's a plethora of study with mice, it's about 2 weeks. By the way, you see this you don't see the behavioral, the phenotype hasn't been studied, but humans, you die earlier. It's about a 26% increase in risk. That's over 3 million subjects, a recent meta-analysis. Fruit flies die earlier, and it's oxidative stress. That's the reason. In humans, we see changes in gene expression for oxidative stress that's in accord. So when I say there's this conservation across various species, I mean, it goes really deep. But social animals, by definition, have sticks and carrots that combine, that lead them to form connections and stay together, form stable bonds. We wouldn't call them social. And some of the consequences of those bonds, when you break them, are similar across these species. So yeah, it varies how long, but in the mice study, it's usually about 2 weeks. Yes.
So besides being blown away by how sophisticated your research is, I was wondering what your plans for the future are. And if you're going to be looking in the future at other types of individual differences and other variables, and if you think there will be different findings based on the specific types of individual differences?
It's a great question. One of the things that NIH has convinced me we need to attend to is doing something about it. I mean, it took me 20 years to convince NIH that there was something seriously wrong here. And then, of course, their immediate response is, okay, now do something about it. And I'm not an intervention researcher, but we worked with the Army and we developed an intervention for them that turned out to work. One of the things that shocked me is loneliness in the Army predicts suicide. So you know the spate of suicides that we've been seeing? And we just finished a paper. I mean, we hadn't even submitted yet, but we just finished this paper where the theory works for Army and non-Army civilians, right? But when you look at how do you operationalize the factors that are relevant, they differ a lot in the Army. In the Army, the collective nature of it makes that particular aspect of loneliness really low. So if I'm going to say in an Army soldier, what's the biggest problem? Where's the biggest source of loneliness? It's not from friends, it's not from spouse, it's from other platoon members. That's where they really suffer. When that goes bad, they have a really difficult time. For civilians, they don't care what their coworkers think. It's what their family, their friends, it shifts. And it's probably because they don't get to choose. In real life, we can avoid coworkers except for when we're at work. In the Army, you're with them 24/7. And so I think that's probably the reason for it. So that then tells us something about what other individuals. However, I have started to look at, because we've been looking at genetics, I have been looking at the big five. And I'll just tell you a snippet. We have about, the heritability of loneliness through behavioral genetics is about 50%. The genetic from common variants is about 17 to 20%. Neuroticism and loneliness share a genetic base. We just did multi-level modeling across lag for the longitudinal study we have, which is a couple hundred people, and the Dutch Twin Register, which is 10,000 people. And in both data sets, we find loneliness leading to changes in neuroticism and vice versa, but the effect of loneliness on neuroticism is actually larger than the effect of neuroticism on loneliness. The other dimensions of the big five are related. They're not responsible. It's really neuroticism that they're sharing. Now, it's not a complete overlap, but there's some shared genetic variants. That raises all kinds of interesting questions about what are those shared processes versus different processes. So that's where we're kind of headed next. Two more questions because I think we're out of time. One more question. I'm sorry.
This is just an observation when it comes to the Army issue. You've got a potentiating factor there because you're dealing with the hypervigilance that goes with the loneliness, activating that implicit hypervigilance, and you're also dealing with the population that's been made hypervigilant by multiple deployments, and even if not exposed to trauma, the kind of vigilance that goes with just being in a combat zone cranks that up, so you've got that potentiating factor there.
Right. And you're absolutely, you talked to vets. There was a general with whom we were, he said he's walking down Chicago streets, he's still looking up at the top of buildings. So you're absolutely right. But it's nice to keep that aspect of these individuals constant so we can look at the effects of loneliness within that. It does change, you're right. Good point. Thank you, everyone. And you have questions, I'm happy to talk to you. Thank you.